Hello, everyone, and thanks so much for joining us for this CV Vault webinar. We're really excited to have Jon Stokes and Jeff Skinner from Stoked Bio here to talk about their AI-driven drug discovery in the antimicrobial and glioblastoma spaces. So, to start us off today, I want to get to know you both a little bit. I'm going to give you a surprise question here. I relate to people through food, so I'm putting you on the spot. And if you could eat one thing right now, what would that thing be to kick us off?
Right now.
Right now, in this
Like
present moment, what's the vibe?
I
For
Oh,
me, yeah, I would go for the old school of chicken wings. I
chicken
do
wings.
enjoy a good
Okay.
bu buffalo chicken wing. Getting ready for the weekend here, getting ready for some fun.
So ranch or blue cheese sauce with the
I
buffalo.
ranch. I have absolutely no interest in blue cheese. You can
Okay,
keep.
okay.
We fight fungus in this company. We don't eat it.
Oh, that's a good tagline. That's great.
I'd say I'm kind of in like a similar camp. Like I'm thinking like a gross pizza. Like not one of those
Ooh,
like fancy
okay.
highfalutin ones like a Domino's or something. Like
A
like
dirty
something that's
pizza.
Ooh,
guaranteed to
yeah.
kill you. But it's like just so self-indulgent and it's like one like you're looking at it and it's like you can't finish the whole thing, but you want to.
Mm,
And you're
And so you
going.
just
yeah,
like keep
yeah,
shoveling.
yeah, yeah.
That
No,
one.
I know
That's what I
exactly.
want.
Okay, okay. So we got chicken wings and pizza with the ranch. I
You know
mean,
why?
John, are
You
you
know why I
stealing some
want
of Jeff's ranch?
no, no, get out of here. And you know why? I'll tell you. So like I have high cholesterol, I found out, and I haven't been eating terribly for like months and months,
Mm.
but like I ate terribly for like 35 years and my body just wants it back.
Mhm.
So like that's why.
Yeah,
You know what I mean?
yeah, yeah. It's just that craving.
Mhm.
Beautiful. Well, maybe for the next Canada UGM, we can
What do
get
you
some
want to
chicken
eat?
wings and
Can't throw that question.
ooh, yeah, okay. It's starting to become fall here in Nashville. Like in the mornings, it's fall, so I'm feeling kind of like a pumpkin pie situation. And I've been playing around with if you powder the pumpkin seeds and put them on top, it's pretty incredible. So I'm thinking I'm moving in the pumpkin pie direction.
All right. A little
Yeah,
fancier than
yeah.
us, but that's okay. Powdered
That's not what
pumpkins.
I was hoping from a guy living in Tennessee. I was
The
hoping for
old
something
school
good.
barbecue.
Yeah, something pumpkin pie. All right, whatever.
I, it's
Yeah, sure.
fall. Don't mess with my don't yuck my yum.
All right, fine, sure.
We love a pumpkin pie, but I mean, yeah, we can have some great barbecue here for sure. But now I guess we can talk about some science instead of just talking about food. So just to frame us from a 10,000-foot view, and this can be to both of you, what is the big problem that Stoked Bio is ultimately trying to solve, and why is now the time to solve it?
Jeff, how about you take a crack at this one?
Sure, absolutely. So we're trying to solve at the core, we're trying to solve resistant diseases. So this is diseases that have developed a defense against the best drugs that we have, and we've run out of options to treat them. It's diseases of high unmet medical need that we absolutely need to find new and novel treatments to bring to the market. And that's really at the core of what we do. We're trying to do the hard things, the things that are difficult to develop, and that we absolutely, as a society and as a medical system, need.
And it's almost like along that same line, like the way that I kind of think of it, it's like things that we will always need. So for example, like I mean, I'm a microbiologist by training, right? So like if we're thinking about antibiotic resistance, right? Every new antibiotic we bring to the market is like very, very, very likely to succumb to resistance at some point, right? So it's like every new antibiotic that we discover has a finite clinical shelf life where it's useful, and then you can't use it because we there's bugs have evolved resistance to it, right? And similarly with like a lot of different types of cancer, right? Like they during treatment they stop responding to treatment because they have changed. So this whole process of, evolution, resulting in our treatments becoming ineffective, it's like we're almost like we're forced to continuously rethink how we're solving these problems because like nature is figuring out solutions to all of our existing solutions, right? And it's like something that I think is cool. And I don't know, Jeff might feel different. It's like we're always forced to like rethink what the best approaches are, because the landscape is constantly shifting under us. Like all these diseases that
Mm,
we're interested in addressing are constantly changing, right? Because resistant
mm,
evolution is happening, right?
Yeah.
mm.
And for me, that's just like super exciting. Like never quite having sure footing ever. For me, it's like a really sort of like, it's like an energizing, exciting place to be.
Yeah, there's been a phrase that's been coming up as the path of least resistance for us. So, and that
Mm,
speaks a couple of things to me. One, we're trying to do things faster, better, cheaper. We're trying to create the path of least resistance to bring novel therapeutics to the market, but we're also trying to create a path where there is as little resistance as possible. So it's been that Stoked Bio is creating the path of least resistance is something
mm.
that's been kind of buzzing around in my head for the last little while.
Yeah, absolutely. So, I mean, that leads perfectly into the next question. I think, John, you kind of already answered this. For you, is it that there's almost like an excitement to the unknown, that these diseases are constantly adjusting, adapting, you need to come up with new solutions. Is that kind of challenge what drew you to antimicrobial resistance and glioblastoma? Like, why did you choose these particular challenges?
So both were complete accidents. Like
Mm,
I genuinely mean that. Like there was no forethought to either one. I
mm,
got into microbiology because I did a fourth year undergraduate thesis in a lab that was awesome, and they just
mm,
happened to be an antibiotics lab. And then as I ended up studying antibiotics, the backstory is I was a fourth year undergraduate thesis in a guy named Eric Brown's lab at McMaster. I started in his lab as a fourth year in September of 2010.
mm,
By the end of September of 2010, I knew I wanted to do a PhD in his lab, like doing antibiotic discovery, just because it was a super cool place to work. Eric was awesome, the group was awesome. And then, once you end up studying some topic for some amount of time, antibiotic resistance in this case, you realize like, oh, this is like a legitimate problem, right? Like a global, like grand challengey type problem. So like, let's talk about AMR for a sec, antimicrobial resistance, and then we'll shift gears to glio. So this year, James, I'm going to throw out a question to you, okay? I'm not going to ask Jeff because he already knows. So how many people this year are going to die from a drug-resistant bacterial infection that we used to treat 20 years ago, no problem?
Oh, oh, I'm very much on the spot. I'm gonna
Oh,
throw out like 10 million.
so 10 million is where we're headed by 2050.
Hmm.
Currently, 1.5 million people a year die from a drug-resistant infection that we used to be able to treat.
Hmm.
And if you do the math, that's one person about every 20 seconds, right?
Oh, wow.
I know it's very frequent, right?
Hmm.
And without immediate action to discover new antibiotics with like legitimate clinical efficacy, like you said, by 2050, it'll be 10 million a year, at which point it's surpassed cancer as a cause of death, right? Um, so there's like an urgent unmet clinical need to address this problem. And as Jeff was alluding to earlier, right, there are like very few, if any, economic incentives to do work in this space, right?
Hmm.
You're going to invest, potentially, let's say $1 billion to make a new antibiotic from soup to nuts. And then these drugs are inherently given to patients for like a week or two or three until they're cured, and then they get to go home, right? So how do you recuperate your investment, right? Go make Ozempic 2.0. You know what I mean?
Yeah.
Um,
Yeah, yeah.
but the fact is like we need antibiotics to uphold the entirety of modern medicine. And let me paint a
Hmm.
picture for you. So if you go in for a surgery, like anything invasive, you're given antibiotics, right? So you don't get an infection. Let's say you got a new hip, you need to be on antibiotics so you don't get an infection in the hip. If you go on, let's say something immunosuppressive, like cancer chemotherapy, you're given antibiotics because you don't have an immune system to fight off bacteria that are living in and on you,
Yeah.
right?
And just on that, so
Um,
people are willing to pay $150,000 a year to treat cancer, but won't pay a buck 50 a dose to treat the disease that'll actually end up killing them.
right, but
Hmm.
and there's historic precedent for that too, right? Like antibiotics have historically been really inexpensive drugs,
Yeah.
right? And they all have a shelf life because resistance is coming because bacteria evolve so quickly, right? So it's like there's all these reasons not to do this, right? But the one reason to do this is if we don't have them, all of these other modern medical procedures that we currently routinely practice also have to stop, right?
Hmm.
So there's nothing like sexy about antibiotics, but it's like they're the foundation of like what all of modern medicine is built on, right?
Hmm.
So it's not really an option. It's like you have the problem, you have to address it, and they're like, that's just that, right? And then the glio stuff, I had coffee with a colleague of mine a couple of years ago. That's how we got into it. I was having a coffee with a super cool colleague named Sheila Singh. She's a neurosurgeon at McMaster, and she runs a brain cancer lab. We had coffee, and she was like, "Do you want to try to use your ML approaches, machine learning approaches, to try to find new drugs to kill glioblastoma?" And I said, "Sure," not knowing anything. But it turned out, as we started working on this project, there's like a lot of sort of like, how do I want to say this? A lot of almost like biological parallels between treating brain cancer and treating a bacterial infection. That actually the two projects kind of like feed off each other. What we learn in glio transfers over to what we learn in antibiotic land, and antibiotics transfer over to glio, right? So it's turned out to be like this like kind of serendipitous, synergistic type of, you know, relationship between these two projects that we're advancing.
Yeah. And just to finish that at full circle, so Jon was in Eric Brown's lab in 2010 as a fourth year student. His office is now beside Eric Brown's in the building that he's in now. So just to have that whole story come around full circle, he's
yeah, dude, yeah, that's
didn't
true.
go
It's
far.
super weird. Like I have to walk past my old lab bench to get to my lab. Like it like when I first it was super trippy, but now it's like
That's how serendipitous is that? That's incredible. Pulling that thread a little more. So, how does the biology first approach for your drug discovery, AI drug discovery, factor into that? Like, what do you mean by biology first when you're describing it? And how does that help you identify and validate the drug candidates you're building?
so let's anchor this in something like super concrete, right? So let's say we want to invent a new drug to treat some nasty infection. Let's say like a really nasty E. coli infection, right? E. coli is like a super common food pathogen. You get sick as a dog, right? Okay. So there's a couple ways that you can think about discovering a drug to treat E. coli infections. The way that I like to think about it is probably the simplest way possible, right? It's we know the thing we're trying to address, which is an E. coli infection. Okay, let's find molecules that kill E. coli, right? As opposed to trying to get overly fancy with it and say like, oh, I think like this protein in E. coli is essential and is going to make a good antibiotic target, yada, yada, yada. You know, we know the phenotype that we're chasing, right? James, if you have an E. coli infection and I'm trying to save you, my goal is to give you a drug that kills the E. coli infection before that drug kills you. That's
That
it.
would be nice.
That's all I'm trying to do, right?
Yeah.
So I already know what I'm optimizing for. I'm trying to find molecules that through some mechanism, and the mechanism I actually don't care right now. I'm just looking for chemical matter that will selectively eliminate E. coli and leave you alone, right? I'm basically inventing a bacterial specific poison, right? So our biology first approach, how we think about this is we know that that is our end use case, right? We want to have a molecule that kills E. coli, leaves you alone, right? So we start to just reverse engineer what this looks like, right? So in a laboratory, we'll go test a whole bunch of chemicals to see which ones kill E. coli in a lab, which ones won't. And then we'll say, well, we know we don't want these molecules to touch you. So we'll also take a whole bunch of human cell lines and test the exact same sets of compounds against all of those to make sure that the molecules that kill E. coli, you know, which ones don't kill human cells too, because we're not trying to find bleach, right? We're not, right? And then we can take all that data. So this is what we call phenotypic data, testing molecules that either inhibit the growth of E. coli or not, and kill or don't a whole bunch of different human cell lines, right? And then we can train AI models on all these chemical structures and their associated activities. Are they antibacterial or are they just anti-life, right? Bleach, right? And then these AI models will then learn to learn how to navigate chemical space in a way that identifies those regions of chemical space that have selective and potent antibacterial agents, right? Now, if you don't care about antibiotics, replace antibiotic with anti-cancer, and it's the exact same problem, right? So you can see that there are a lot of parallels between selectively targeting one specific type of cell in a whole bunch of cells that define James, right?
Yeah.
So that's
Yeah.
what we mean by biology first. Our ML models and all that fancy stuff we do in the computer is simply, a powerful set of tools to help us explore the vastness of chemical space to identify those molecules that are most likely to have the therapeutically relevant properties that we want, right?
Mm.
We're not like fully leaning on the ML, like just for the sake of it. We're using it as, a tool. Like let me put it this way. Do you ever like walk into a lab and have someone brag about their pipettes? Do you?
I don't know if that happened to Vandy.
Yeah,
That
yeah,
hasn't
yeah.
happened to
We
me,
do.
right?
Yeah, we do PCR better than anyone else.
So like that's how I think about all of these like fancy ML models that we always employ, right? Like they're just really cool pipettes that help us do more experiments in silico than we could in the lab, right? But the success of those ML approaches hinges on the quantity and the quality and the biological relevance of the data on which they were trained, right?
Mm.
So us
Mm.
being like what I'll call domain experts. I hate that word. I actually don't know what an expert is. It's a stupid word. But like we know a thing or two about bacteria, right? We know a thing or two about brain cancer, right? And we know a thing or two about like what we want these different types of molecules to do when someone eventually eats them, right?
Mm.
So we can begin to bake in all of these properties that we want really early in our ML modeling pipeline to maximize the probability that when those ML models output a new chemical, here it is, that it's likely to have all the properties we want towards like translational development. You know what I mean?
Mm.
Did that make any
Yeah,
sense?
and no, no, that makes total sense. And you've brought up in the past that you're working in a space that hasn't traditionally been successful for big pharma, right? Because the points you brought up, you find your new drug, you spend millions of dollars making this drug, and it goes on a shelf. So do you see this AI approach bringing the cost down enough so that this is going to be a successful business model? Is that one of the angles? And maybe Jeff, that might be
Yeah.
for you.
Yeah, I mean, it's not going to save as much time as we hope because you still have to go through all the clinical pathway. So you're looking at cutting, from the discovery or the early stage, you're looking at cutting 2, 3, maybe 4 years off the development timeline. But in
Mm.
terms of cost, you can easily cut, 40 to 50% of the cost of a drug because of how many drugs you need to start with. And it's the classic one. For every drug that you get approved, you have to find 10,000 compounds. And at some point along the way, you have to kill that drug. And any dollar that you spend on that one that dies is money that's just lost. And you're never going to get you're never going to sell it. You're never going to do anything with it. So by doing it this way, by trying to find earlier, faster compounds that have been trained on this biology approach, we have the potential to save a significant amount of money by eliminating the number of failures as we continue to move forward. And I mean, there will still be failures, and we're still seeing failures in phase 2 and phase 3, but it's really trying to eliminate the amount of money that we spend on the preclinical development, which can be vast amounts of money for all the failures that we see.
Mm. Absolutely, especially for early stage. I think that
Correct.
that can be incredibly powerful, right? Um,
That was Jeff's like measured answer. I'll tell you what, like I think we might be able to do. I'm hoping. I want
oh
it like
yeah.
so what I always hope for is like twice as fast, half as much, right? Do stuff, get a drug to market twice as fast as traditional and make it cost half as much money. That's sort of like where I've positioned sort of like our first goalpost. You know what I mean? And then once you do that, where's the next goalpost? Well, let's do it again. Half as much, twice as fast, right? Because I think like as these both the models themselves, as well as asterisk potentially more importantly, potentially, I don't know, the data sets that we curate to train these models improve, right? You can kind of see this happening like faster and faster and faster with every success. You know what I mean? And also faster and faster and faster with every failure too, because like there's a whole bunch. It's like we're learning the ways not to do this too, right? So I think like our first, I'll say our read that as like my sort of first like mental goalpost is like, can we invent a drug from idea to giving it to a patient or like a bunch of patients because it's been approved, right? For half as much money and twice as fast as everyone else. To me, that'd be cool. I know it's lofty and it makes me sound like arrogant or whatever, but I think we can do it. You know
Well,
what I
bigger transformational changes have happened. It's not that there are precedents in science, right? I mean, think shotgun sequencing. Before shotgun sequencing came out, it was ridiculously expensive to try and march through the genome, and then new technology comes out, and it's just like, eh.
mean? Yeah,
Now we can sequence your dog's genome, like figure out if
yeah,
they're schnauzer or not, you know?
yeah, yeah, yeah. Yeah, no, I
So
feel
I
that.
love a lofty goal. I mean, kind of pulling that thread a little more, getting kind of into some of the specific models, some of the specific platforms. Can you talk a little bit specifically about the MOSAIC platform and how that works on your lead compound generation and what makes it unique, kind of in the field of recurrent glioblastoma?
Sure. And so MOSAIC is a Stoked Bio brand. So it's basically taking what Jon does and putting a fancy name on it, modeling structures for anti-infective and cancer, a brand. And I've spent a lot of time looking external to find out if, who else is doing this the same way that Jon does. And I don't know if Jon can speak to it. I've not found anybody who's doing this the same way. So it makes you the process that Jon from ideation to delivering of a compound with proof of concept, a very, very unique process. And so that for me, that deserved a name. So I came up with MOSAIC. And the idea of MOSAIC is, a mosaic is a whole bunch of pie. You start with a pile of tiles, you do something with it, and you end up with a picture. Right. And so that kind of from the data to a compound kind of made sense to me that this was kind of putting together a mosaic and ending up with a compound and a treatment. But I mean, for the process, and Jon can get into the nuts and bolts and the details a lot more. But this started with an idea. This started with Jon and the collaborator Dr. Singh out of McMaster University having a conversation, and Sheila's a fantastic she's a brain surgeon. She actually treats patients with glioblastoma, and she created a biobank, essentially taking some samples of every tumor that she removed and kept it in a fridge. And so the conversation was, if you had access to these tumors, to these cells, could you do something with that? Could you generate some compounds that could potentially end up treating it? And that's basically the starting point of MOSAIC, or the starting point of Jon's process is, a selection of a disease, and then isolation or delivery of cells or tumors or something that you can treat from that. And so that's where the idea, I think, for the glioblastoma, we talked about a little bit about being serendipitous, where it came from. But it was that access point to have the disease of interest. And so what's really unique about Jon's process is the next step. Once you have those cells, that disease state, Jon treats them in such a way that the training data comes out is the biological response of a vast chemical space on that disease of interest, on that specific disease of interest. The training data basically has what kills it and what doesn't kill it, what characteristics of chemicals kills it, what characteristics leaves it alone. And that's what leads into the models, and why the models can be so powerful, because it has this unique and proprietary and powerful training data that it can then chew on and look into a vast chemical space to find novel therapeutics to treat. And then the MOSAIC, again, the whole process, it isn't done there. A lot of the in silico, a lot of our competitors stop there and say, here's a long list of chemicals that the models say work. Jon filters those down through preset characteristics to say, this has the potential to be a drug. This has potential to get into the brain, past the blood-brain barrier, for example, for glioblastoma. All of these ADMET and toxicity characteristics that we can filter the compounds down, and output on there is, again, a smaller but powerful list of compounds, which then Jon manufactures and then tests in biological system. And that's all done, and all of that step is done, and even potentially iterative steps based on the biological data that is generated in Jon's lab, going back into the models to strengthen the models and output even better molecules that can treat the disease. And so it's as powerful from biology into the computer and then back into biology to test to make sure the computer is doing, or the compounds are doing what the models say they should be doing. And that whole process that as I said, Stoked Bio is branded as MOSAIC, and it's basically what Jon has created. He has a habit of underselling himself and it being super easy and super chill. Maybe for him it is, but it is an incredibly elegant process that he's created. That, as I said, is biology first and biology last, which I think is just as important.
Can you tell
And it
that
sounds,
he's in
oh, go ahead.
pitches?
Yeah,
Like
I'm doing pitch competitions. I have 3, 5, 8 minutes, and so I've got these sound bites down.
the answer that I've given is like, I don't know. We just kind of do it.
This is why we work together. This is why we collaborate. I think so that's a perfect segue. I mean, you've talked about generating a lot of compounds, a lot of data, and you're, collaborating on these projects and working with the team. So what is a way, when you're managing all these collaborations, how do you stay consistent? How do you, have a single source of truth, especially when you're working with multiple groups across like a giant pipeline where a lot of data is being processed and generated.
What do
At
you
this point,
mean?
before John says anything, I'm going to say CDD Vault is the best way to do all of
Excellent.
that.
Excellent.
John, you can expand on that now.
That's true, actually. Yeah, like I'm not blowing smoke. Like we just set up, like my lab just set up its internal vault, and like people are using like they're like the uptake is way quicker than I thought it was going to be. You know what I mean? Because we have a million projects going on. I don't know how many I've lost track of how many like compounds that we're looking at for various therapeutic indications. And like before, all of our data was stored on like napkins. You know what I mean? And like written on people's hands and stuff. And like, yeah, like
He's,
having it in one
he's,
place is actually, I think
he's
it's been
not kidding.
I'm not, I'm like, you think I'm not joking. Like there were like you'd see like stacks of like paper towels with stuff written on it on our benches. And I was like, there has to be a better way. Um, but honestly, dude, like when it comes to, maintaining, what was the word you used? Consistency or something?
Hmm.
Like across all these projects. I mean, like everybody knows what good science looks like, right? And like what it looks like if you're trying to cut a corner. Like the the principles of like running a super robust, drug discovery project in antibiotics is the same as if we're doing brain cancer or antiviral or antifungal or whatever, right? You know, it always stems from like, do you have like the most robust biologically relevant training data set that you can get, right? Within the confines of your lab and your budget and whatever, right? And then like once you have that data, it's like, what is the best way, or is there a way that I should be incorporating some ML guided pipeline to help me maximize the capabilities of this chemical screen that I've done to address some therapeutic indication, right? Like the principles remain the same. The nuts and bolts and like what specific type of training data, like what does your training data look like? What does your model architecture look like? And all like that type of stuff will change. And that's highly dependent on the specific task at hand, right? But at 30,000 feet, like you know what good science looks like. You know what I mean? So I don't know. We try not to overcomplicate this stuff. We always go back to the basics, right? Like, we're trying to discover a drug to address X. Okay, how can we go about gathering the best, most relevant training data to train a model that will help us find drugs to address X? Right? This
And
isn't,
ideally
this
not
isn't
stored
like,
on
dude, this
notes
isn't
on
rocket science,
on
right?
paper towel.
Like we're
Yeah,
not
yeah,
building the
exactly.
ISS here, right?
Yeah.
Like, I don't know. It like to me, it's like just strip away all the complexity and like what are you left with? That's what you need to focus on, right? And the
Hmm.
complexity will
Hmm.
come. Like, well, I always say, like, when we start on drug discovery projects, right, regardless of what it is, whether it's like an antibiotic, a glio, we're working on like like we're working on a crazy antiviral project right now, like an antiviral drug discovery project. I can't get into it, but it's rad. Like we always start from the same place, right? It's like, what does the best training data look like? Right.
Hmm.
And that's like, again, going back to this like biology first, right? What does the best training data look like? So a model will output molecules that do the thing we want it to do when we try to translate this into a patient. Right. So
Absolutely. No, and I really appreciate you saying that. And I'm glad that CED Vault can help you in the work that's so important, so impactful in the current landscape we live in. Antibiotic resistance is a impending catastrophe, so I'm glad we can support you in that.
well, I mean, yeah, it's
Uh,
like I'm glad that stuff isn't stored on like in like napkins and Sharpies and stuff. I mean,
the
honestly, if I'm
I'm
honest,
saying
it probably still is, but at least like the idea of having a place where we could theoretically store stuff is comforting. You know what I mean?
No, yeah, it will eventually get back to, it doesn't stay on the paper napkins. That's an intermediate state before it gets added to
It
the
might be
ground
like it
truth.
might be like an inherent filtering state. If like if the stuff on the napkins gets thrown out, it was probably not important anyway. Or at least I hope.
If
I
you could
mean,
add that feature to CD Vault, a napkin scan, I think you would take over a lot more labs in at universities.
We can get
oh,
that we
dude,
can
you
get
can
that to the
imagine,
dev team
right? Like all it
immediately.
like scribbles on napkins and like random pieces of paper. If someone can just snap it with their phone, and then it just like
You
magically goes somewhere.
Yeah,
you
Josh,
can add
we're giving
pictures
you
to the
yeah,
ELN.
yeah. We're giving you that task. We expect to be done next week.
Absolutely. We'll get that through the pipeline. We'll push it through, cancel everything else. Uh, so kind of popping back out to 10,000 foot view, kind of coming to the foundational story, the team culture that you're building at Stoked Bio and the vision. So Jon, you had a successful academic lab before launching Stoked Bio. So what led you to take this leap into entrepreneurship, and what would you say was the founding moment for Stoked Bio?
He was
Yeah,
tricked and he was forced.
I was tricked into it. I'm not joking. Like, and you say like successful academic lab, like it's like, and you said it's like like it's not my lab. Like I'll be the first to say like I could get hit by a bus like in an hour, and that lab is still going to be totally fine. You know what I mean? It's like, and I genuinely mean that. Like Jeff knows everybody in my lab. Like they don't need me for anything. Like they're all way smarter and like just way better than I am. So yeah, whatever. But it's true.
It sounds like you set up a really good lab.
Listen, people are good. I don't know what to say. Um, but like, so yeah, I mean, I've always wanted to be a professor for as long as I could, like a scientist who just like gets to do their own thing. Like what I like about academia, it's like I have like bosses on paper, but like not really. You know, nobody can really tell me what to do, which is like super liberating. Like I love it, right? Um, but you know, it like it's obvious. Like we're like basically a preclinical like drug discovery laboratory. That's what I like to do, right? Figure out new ways to find molecules that have therapeutic potential for like unmet stuff that lots of people aren't working on, right? Because something I was always taught, it's like if everybody zigs, you zag, right?
Mhm.
So nobody's doing antibiotics really. Nobody's doing a ton of glio stuff really. You know what I mean? So it's like I kind of like it's cool. I have like a playground. There's some people, but like I don't whatever. I'm not bumping into a million people, let's say. So I was at the Stoke Bio's co-founder's house um for dinner a couple of years ago, I guess. And his name is Chris Sinclair, and he's like a successful entrepreneur in and of himself. He has like a branded merchandise company that's made him a bunch of money. You know, it's like you know sweaters that have like a company logo on them. That's what he makes and sells, and like pens and stuff. Uh, he's made a bunch of money off that. So I was at his house a couple of years ago, and I was telling him, explaining to him what we did in the lab, and he was like, "Oh, this is like, why aren't you monetizing this? Like trying to do something useful, right? As opposed to just like writing papers and who cares?" And I was like, "Well, I care. I think papers are cool. I read." But he's like, "Yeah, but like 99% of the world doesn't. So like, what are you doing?" And I was like, "Oh yeah, that's a good point, blah blah blah." Um, like a month later, he just sort of made this company. I'm not joking. That's how it happened. He's like, you know, this company exists now. Here's a bank account. Figure it out, right? And then I was like, I don't have the bandwidth to figure this out. I'm a scientist. I don't know anything about this whole businessy stuff. And he said, all right, leave it to me. And then a little bit after that, I get an email from Jeff saying, like, hey, I'm running this company now. Right. Like that's kind of how this worked, right? And it was super organic. And that's what I loved about this. Nothing was forced. Not really, right?
Right.
Um, and what was also super cool is like, you know, I got to know Jeff and I realized like Jeff knows what he's doing, right? He's like a 30-year like pharma like veteran, right? And what was rad is like because Jeff's running the business stuff, like I can just focus on science. Like I'm doing all the stuff I was gonna do anyway. right? But what's really cool now is like the relationship that my lab has with Stoked, and by extension, the relationship, like the super close, really positive relationship that my university has with Stoked, right? It's like we have this like what I think is like a seamless intellectual continuity between the stuff that we're inventing in the laboratory and the stuff that the company is I'm going to use a very specific word here, innovating, i.e., turning an invention into a product, right? You have like an unbroken line of continuity, right? And that makes this transfer of intellectual property and ideas, right, from the academic lab to the company as a translation engine, like perfectly seamless, right? And again, that's like another opportunity to increase the efficiency with which we can translate academic discoveries into things that actually help people. You know what I mean?
Yep.
And like the fact
Yeah.
that this all happened sort of like by accident makes it all the more like perfect for me, right? Because there's nothing forced. No, I'm not twisting anybody's arm to do this. You know what I mean?
Yeah. Yeah, and it was the perfect overlap with the skill set. So Jon is a scientist. That's all he wants to do. Chris is an entrepreneur, been very successful at that. And then myself, I've had, as Jon said, not quite 30 to 25 years in pharma and biotech in Canada, mainly in business development and executive management. But that means I have a basic understanding of the industry as well as know a lot of people within the industry. So there's no overlap around that. And we just hired our first couple of people, the one that started in August, who came out of the CDL program at Rotman's. Great kid who's really really smart, guy named Steven. He was asking about the culture that we're bringing into, and as you can see from Jon, hopefully you can see from myself, and Chris is the same. There's no ego on this. We're here to solve a problem, and if we can help to do that, we want to be involved. If Chris thinks that he can help, he's there. He's the first one there. If he can't, he steps back and lets Jon and I handle it. If Jon is lost in the science and is doing that and doing the stuff that he loves, and Chris and I need to make a business decision, we do it. We let Jon know, but Jon isn't going to, why wasn't I involved? He trusts us. He's happy to continue in the lab and to build that. So it's this great relationship that we're developing. And then the added thing as well is that we're trying to do this. I'm not going to say on a shoestring, but in a way that's resource constrained. Jon's lab is always resource constrained. Biotech as an industry is always resource constrained, and I think that creates smarter ways to do things, and maybe not innovation, but better ways to bring this out on a smaller budget, but have this big, huge impact that we can all be really excited about. So it was a wonderful opportunity. I met Chris through another friend of mine who's in the brand and merchandise business, and as I said, it just kind of flowed organically from there, and it's just been a wonderful ride so far.
That's what I like. One of the coolest things about this, it's like, I mean, I love it. I know you don't love it. We've had this conversation. It's like, how much can you do with like as few dollars as possible? Like you're forced to like really, really think about every decision and be like super creative at like every key decision making step, right? Like, I don't know. Like to me, it seems like I'm not going to mention any names, right? But like there are biotech companies, like early stage biotech companies that get flooded with venture cash, and then they just make a ton of terrible decisions, right? Because like they're swimming in money for a while. And it's that's just like so like financially irresponsible, both to yourself, the patients who you're hoping to serve, and the investors who gave you their money. Like I've never, like, I just don't understand that philosophy, right? It's like I work much more effectively when it's like I know I all like it's like almost I don't have quite enough, right? So, and like by not ever having quite enough, like you just have to be really sort of nimble. You know what I mean? And to me, that's like
Yeah, I
part
mean,
of this fun game. Like I love that, right?
that's the perfect. So the next question was going to be like, you want Stoked Bio to be the best in the world, and I think that's a perfect segue. Like, how are you making Stoked Bio the best in the world at what it does, culturally, scientifically, operationally? When you have these values that you're holding, like, how are you deploying that?
Yeah, I mean, it starts with the product. So we're doing things that the world actually needs, healthcare actually needs. So we're building that, and if it's hard, that's even better. So we're trying to build that culture into that we do the things that the medical systems that that science needs. and we do the things that are hard to do. One of the things is a core for us, and we want to continue to do, and we've been able to successfully do that. Is we want to remain Canadian. That is a big. We want to remain in Hamilton. I mean, if you get even more specific on that, we want to build a company that the city, that the province, and the country can be proud of, and do that in a way that is bringing in Canadian investors. So far we are raising money, and we've been able to bring in almost every dollar from Canada, which is not an easy thing to do, and we want to continue to do that. We have the ideals in here that have been kind of ground into us by our Canadian backgrounds, and we hope to continue to build that.
Yeah. And again, it's like always focusing on like what are you trying to do, like actually like get rid of all of the complexity, right? Going back to like the individual patient you're trying to serve, right? Or the population of patients you're trying to serve, right? And for brain cancer, it's like, okay, we have these people getting glioblastoma, they undergo brain surgery, someone gets a spoon and scoops it out, then they get temozolomide and radiation, and then it comes back and then they die. And that happens in 12 months, what I just described, 12 to 15 months, whatever. It's like these patients don't have options right now. It's a remarkably difficult problem to solve. But I think, the way that we like to think about this, again, not focusing on targets or any of that fancy biology stuff, is like going down way back to basics. Can I kill a cancer cell selectively? Right?
Mm-hmm.
Can I kill a bacterium selectively? Can I eliminate a virus selectively while leaving all of the healthy human stuff alone? That like very simple idea drives a lot of what we do, right? And it's like a lot of times it's easy to get caught up in the complexity of whatever you're working with. You know what I mean? And it's like it's hard for us sometimes to not get lost in the sauce and take a big step back and say, hey, is the thing we're doing right now, the thing that's right in front of us, the direct next step to getting a drug into a sick patient, right? So I don't know. And maybe other people think like that. I don't know, but like that's how we think. And at least that's how I think. I hope that's how other people think too. But yeah, I mean, it's like, and we're just like Canadians. I don't know. I
Yeah.
like being Canadian. I'm sorry.
Yes.
James, are
No.
you
Oh,
American?
yes, he just apologized for it because he liked being Canadian. There's nothing more Canadian in the world than
And the ego
that.
thing is
Maybe.
here too. Like what I like about like sort of the culture that we're building at the company, it's like you never have to say you're sorry. You know what I mean? Because like, everything we do, every criticism, every compliment, whatever, like it comes from a place of like caring deeply about what we're all trying to do. And also it comes from a place of like deep trust and respect too. Like I can say anything to Jeff, anything in the world, and I don't have to say I'm sorry because I know he's fine with it, and vice versa. And with
Yep.
Chris and with Steve and with everybody in my lab, right? Like we're all just like we get it, right?
Yep.
And the fact that we can be super open and honest with each other, science aside, is just like sort of like the foundation of efficient progress, right? Because no one's guarded, no one's like hiding anything. No one's like, oh, I don't want to talk about this. You know what I mean?
Yeah, yeah. So you be the best by letting everyone be the best version of
Yeah.
themselves and genuine selves in an
And
Yeah.
environment where you,
let me
yeah.
brag too. Like when it comes to like what we do, like our science, I'll go toe to toe with anybody. You know what I mean? Like we can cook. So you know what I mean? So I'm
Yep.
not like yeah, it is what it is.
Absolutely. Yeah. Okay, so I'm excited. So last question, and based on the conversation, I'm so excited. What are you most optimistic about? Whether scientific progress or commercial partnerships, like what is on the horizon that you are stoked about?
Oh, stoked. I like it. Very, very good. That's
Dude,
what I'm
James
most
had
excited
that
about.
ready to go. He was
I'm
waiting
done.
for
I can
I
leave.
was waiting for you. He had it loaded up. I'm most excited that everybody's going to be stoked about something. From my side, and again, it's going to be very different from John, but it'll be when we get that first partnership from pharma that are realizing that the science is, because we know everybody that is here and that is doing this daily. We know how good and how pure the science is that John is doing, and then by extension of that, and John will say that the kids in his lab are doing. So we all know what the quality of the stuff is out there. We need to convince others. So I'm looking forward to, and what I'm most stoked about is, again, from my background and what I've done for my entire career, is create partnerships, create relationships. So I'm really excited to take the first molecule that gets to a stage that has enough of a data package, that has enough information to go in front of pharma and say, look, this is what we're doing in the lab. Either take the one that we've already developed, or work with us to develop one that you're really excited about yourself. I mean, again, from the business development, it doesn't get any better than that. It's a lot of fun. Now, Jon's going to talk about the science a little bit more, and that's what he should be doing. And call me a nerd for being excited about doing a pharma partnership, but that's what I am.
I wasn't gonna call you a nerd. I was gonna be like, that's like the most business person answer ever.
Thank you.
I'm excited about my first pharma partnership.
Yes. Yep.
So
I
I
should
have
have
three
done.
things, right?
I
That
should have done that with a bow tie.
I know you should have. There's three things kind of that I'm like, that I think about a lot. That, like, where I'd be like, okay, I've done something like good. You know what I mean? Like, like when you did something okay, you get like that feeling in your tummy. Like, and like you're like everything kind of just sinks and you're like, okay, like
Yeah.
So
The
there's three.
Domino's pizza effect.
I mean, the Domino's pizza effect is like down, but you feel awful about yourself. It's like
Yeah.
self-loathing. You know what I mean? Where you're just like covered in pizza grease.
Yeah, with the pizza baby trying to burst its way out.
Like, no, but like it's like, so there's three things. So the first one is first, like the first molecule that comes out of our group that goes first in human, that'll be like a like one, what's it called? What's like in your life you have like these like moments. What are they called?
Epiphanies?
Nope.
No.
There's like not
Transformation?
landmark. The word will come to me, but that'll be like,
Milestones?
I'll put a pin in that in my timeline. You know what I mean? The next one is when something that we had hands on gets approved for patients like to use around the world. And the third one that I'm really excited about is when I've made all of my students and people who work here way richer than me. And I mean that. I genuinely mean that, right? Like when everybody who has worked sort of in this ecosystem can do whatever they want, right? Because of the work that we have collectively done, then I got the A+ there too, right?
Yeah.
So it's first in human, first approval, and then everybody here can help me to do whatever they want because they don't care.
And I had a mild, I had a much smaller thing like that. So I worked for a generic pharmaceutical company. We worked hard to bring in a number of generic drugs into the market. A few of them that were rare, a few of them that were difficult to get. And the first time somebody that I knew received the drug that I was responsible for bringing into Canada, that was a really cool feeling. And that's not even close to inventing a drug and seeing it go all the way to the end and getting it into the patients. And Jon and I both have daughters. To be able to say at one point to our daughters, that person is alive because of something we had a hand in, I think would be an amazing day.
Yeah.
Absolutely.
No
That
pressure,
sounds
right? Easy.
Sure. Yeah. I'm expecting it by Christmas.
Oh, Q3, we're still in
Yeah.
it. Yeah. Oh, well, thank you so much, Jon. This has been a delightful conversation, and I'm so excited to see Stoked Bio go forth and change the world. And I think, fantastic team, fantastic tech, and looking forward to you guys changing the world. So that leads us to the end of the webinar, displaying Jon's email. If you want to reach out to him directly with more questions, you can get in touch there. And we'll also share links so that you can get in touch with the organization's website and social media and get in touch on your own. So now we're going to move into the Q&A. We just have a couple questions that came up. So how does the MOSAIC platform integrate generative AI outputs into experimental workflows?
Jeff, you want to go?
Nope.
Okay. Yeah, so I'll say this, like generative AI for like drug discovery can mean many things. right? So I'll explain to you how we implement it, and then I'll explain to you how like it sort of feeds our experimental pipeline. So as sort of we discussed, right? Like we'll gather, let's anchor this in something E. coli again. Let's come back to E. coli. I love E. coli. During my whole PhD, all I did was pipette E. coli every day. So I love E. coli. All right, E. coli, we want to kill it. Gather a chemical training data set in a laboratory, go train some ML model that will design new antibiotics. Sounds like a good idea, right? Okay. The idea of designing molecules to do something using an ML model is like old, like pretty old, like 20 years, maybe more probably. But here's the thing, the overwhelming majority of these models design molecules atom by atom, which sounds good on paper, right? Like you can build like any chemically valid molecule. But you know what the issue is? What's the issue? I have my professor hat on. James, what's the issue?
I'm a biologist. You can't put me on the spot
You can't
there.
make them, right? The
Okay, yeah.
overwhelming majority of molecules that are outputted by generative models that build molecules atom by atom are synthetically
Mm.
intractable, which means you
Mm.
show them to your chemist buddy down the hall and they say, get out, right? Either
And
because
that's literally, and he's literally down the hall from
that's
John.
literally happened. And it's because either the synthetic technologies don't exist to make the molecule or the yields are terrible and it's super expensive, right?
Mm.
So all of our generative models that we are currently developing and deploying for the company they don't work atom by atom. They work by molecular fragment. So what we've done is amassed a really big bucket of multi-atomic molecular fragments. These are like, think of them like Lego bricks. And then beside the big bucket, we have another bucket of a whole bunch of reaction schemes, right? So we know how every building block reacts with every other building block across all these different reactions. So what we can now do is tell our generative model to go building block by building block and reaction by reaction, sticking different building blocks together in different orientations to build full molecules that have all of the properties we want, right?
Mm.
So currently, like as we are speaking now, we work in a synthetically accessible chemical space of about 150,000 building blocks and 50 synthesis reactions. And that gives us access to about 46 billion chemicals that are synthesizable in 1 or 2 steps. And what's super cool is I can get every molecule for about 200 bucks. So no money, right? So we've turned this like, how do we search like the vastness of chemical space and from an atom by atom problem where you inevitably can't make the overwhelming majority of stuff that you output to this super practical tractable problem by building molecules using Lego bricks, right? So because we have this generative framework where we know that most of the stuff that is designed, we can synthesize super easy in the lab, either ourselves or with collaborators or with like a CRO. That means that we can generate a bunch of stuff, synthesize a bunch of stuff, bring a bunch of stuff into the lab and see what worked and what didn't, right? We're not left in this spot where, oh, we've generated 100 molecules, we can only synthesize 2, right?
Mm.
We're left in a spot where, oh, we generated 100 molecules that look really, really promising. We were able to synthesize 90, right?
Mm.
And by being able to amass that quantity of like AI predicted data in the laboratory, see what worked, what didn't, and then feed it back into the model, that just increases, the rate at which these models learn to identify novel molecules that
Mm.
satisfy all these properties, right?
Mm.
So like, yeah, again, it's like, I like how Jeff said it earlier. It's like biology first, biology last. You know what I
Mm.
mean? Like it's always the biology that's informing the model. You know what I mean?
Absolutely. Yeah, that's fantastic. I really appreciate you breaking that down. So that does unfortunately bring us to the end of today's webinar. We'll get the rest of the questions answered by email. So thank you so much to the audience for joining us today. And John and Jeff, it has been a pleasure to chat with you and learn about Stoked Bio. And I hope everyone has a great rest of your day. Thanks so much and take care.
Hi, everybody.
Bye.
Everyone, thank you.